Hello everyone and welcome to my channel. Here we are discussing the classification of viruses, Baltimore classification, LHT system classification, and a classification given by a ICTV. So subscribe my channel Easy Life Science, like my video and share it to your friends. Press the bell icon so that you will get a notification time to time. So let's start. Virus classification. For a classification of viruses into a particular taxa, the viruses were studied a number of characteristics. They were studied for their phenotypic characters, their mode of replication, in which host they replicate, and which kind of disease they produces. Also, in which organ or which target organ they replicate or they causes a pathogenicity. Such characteristics were used for a traditional classification. In 1972, a scientist Johnson, he named the viruses on the basis of the disease caused by them. For example, the polio virus, mumps virus, influenza virus, rabies virus. These viruses are named on the basis of their disease. Also, the bacteriophages were named after a laboratory code symbols. For example, 5X174, M13, 56 QB virus and T4 bacteriophages. These are laboratory code symbols and these are given to names of bacteriophages. The virus classification has four classification systems. One is a Holmes classification, second is a LHT system classification, Baltimore and ICTV. The first, that is a Holmes classification. In 1948, the Holmes used a linear system of binomial nomenclature and he classified viruses into an order Virales, and the order Virales further sub-classify into a suborders, that is a Phagini, Phytophagini and Zoophagini. The Phagini, these are viruses which attacks a bacteria. In a group first or in a Phagini suborder, the Holmes classify all the bacteriophages. Then the suborder or a second suborder, that is a Phytophagini. Here the scientist Holmes classify all the plant viruses, and in group third or a suborder third, that is Zoophagini, Holmes classify all the animal viruses. And for this classification, the scientist Holmes used up a Carl's Linnaeus binomial nomenclature system. Here there is a use of two words for calling a name of virus. Then the LHT system of a classification. In 1962, three scientists, the Loff, Horney and Tournier, three scientists were there, L for Loff, H for Horney and T for Tournier. So it becomes a LHT system and it is given by in 1962, and this system gives a advanced scheme for a classification of virus, and this system grouped viruses into a phylum Vira, class Ribovira or a Deoxyvira, order Virales, family Viridae, subfamily Virinae, genus ends with a name of virus and a species. And the International Committee on a Nomenclature of Viruses, that is ICNV, it was set in 1966 at International Congress for Microbiology, which were held in Moscow. And this International Congress for Microbiology accepted many principles of ICNV. And in 1933, The ICNV is replaced by another name, that is ICTV, International Committee on Taxonomy of Viruses. The LHT system used a number of criteria for a classification of virus. The first criteria, that was a nature of nucleic acid, whether it is DNA or RNA. The second criteria was symmetry of a capsid, whether it is icosahedral, helical, or it is complex. Third criteria, presence or absence of a lipid envelope around a capsid. And the fourth criteria, that was dimensions of a virion and a capsid. How much capsomers are there? Then the fifth criteria were how the capsid get formed around the nucleus, nuclear material. These were the criteria used by the LHT system for a viral classification, and this classification was approved by one committee named as a PNVC, Provisional Committee on a Nomenclature of Viruses. The LHT system, it starts with a phylum Vira. All the viruses are grouped in a phylum, that is a Vira, and this phylum is subdivided into two subphyla. The first subphylum, that is a Deoxyvira, and second was a Ribovira. In first subphylum, all the DNA viruses are included. Deoxyvira, it is a subphylum, it contains all the DNA viruses. It is further divided into three classes: Deoxybinala, Deoxyhelica, Deoxycubica. In Deoxybinala, DNA containing complex symmetry viruses were kept. It has one order, that is Urovirales, family of Phagoviridae. All the bacteriophages are kept under this family Phagoviridae. The characteristic of this family was, these are complex capsid symmetry DNA containing viruses. Second class, that is Deoxyhelica, and DNA containing helical capsid symmetry viruses are put into this class. It has one order, that is Chitovirales, family Poxviridae. Then third, that is class Deoxycubica. In this class, the capsid symmetry, that is the icosahedral capsid symmetry viruses are kept. It has two orders, one is a Peplovirales and another is a Haplovirales. The Haplovirales order, in this order naked viruses are kept, whereas in Peplovirales mantle viruses or enveloped viruses are kept, and these were all icosahedral viruses which contain DNA as their genetic material. The Peplovirales or mantle viruses having envelope around their capsid, and capsid is a icosahedral. It has a family Herpesviridae. The Herpesviridae family is characterized by a presence of 162 number of capsomers. Then the order Haplovirales, it contains naked viruses, and it is further subdivided into 5 different family. The family Iridoviridae, the capsid of viruses undergoing this family contains 812 capsomers, and these are also called as insect viruses. The next family, that is Adenoviridae. In this Adenoviridae family, the viruses are kept having icosahedral symmetry, capsid is without envelope and it contains DNA as their genetic material, and the capsid is made up of 252 capsomers. Example, adenoviruses. Then third, Papilloviridae. The Papilloviridae family, their capsid is made up of 72 number of capsomers. Example of a member of this family is a Peplovirales. Then the fourth family, Parvoviridae. Viruses of this family shows a capsid which is made up of 32 number of capsomers. Then the fifth family, that is a Microbiological. It contains a viruses with a capsid having a 12 number of capsomers. These are the two orders, Peplovirales and Haplovirales, and these orders are a sub classification of a class Deoxycubica. Then the subphylum Ribovira, we seen a subphylum Deoxyvira where there were all the DNA viruses, but in subphylum Ribovira RNA viruses are kept, and it is subclassified into two classes. One is Ribocubica and another one is Ribohelica. The Ribocubica, these viruses having a cubical symmetry or a icosahedral symmetry. It is with two orders, Togavirales and Limovirales. The Togavirales having one family Arboviridae, whereas Limovirales order, it is with two families, Nepoviridae and Rivoviridae. Then the second class of a Ribovira, that is a Ribohelica. Helical symmetry viruses are kept under this class. It is further classified into a order Phagovirales, and the Phagovirales having a three family, Myxoviridae, Paramyxoviridae and Stomataviridae. Here plant viruses are classified, Stomataviridae, Paramyxoviridae and Myxoviridae. In this family there were a classification of plant viruses, and they undergoing order, that is a Phagovirales. The next order of Ribohelica, that is Rhabdovirales. It has further two suborders. One is the Phagoviridae and second is Rigidovirales. In both the suborders there were a keeping of plant viruses. The suborder Phagoviridae is having two families, Mesoviridae and Peploviridae. Then the suborder Rigidovirales further split into three families, Pachyviridae, Protoviridae and Dolichoviridae. Here you can see for a DNA viruses or a classification of phylum Vira to the subphylum Deoxyvira, Parvo, Papo, Adeno, Hepatidine, Herpes, Irido, Buccalo and Fox. You can take a screenshot of it, and in this image you can see for a subphylum Ribovira. Take a screenshot of it. Then next system of a viral classification, that is a Baltimore classification, and it is given by a scientist David Baltimore in 1971. And according to David Baltimore, he classified the viruses into 7 groups on the basis of type of nucleic acid, whether it is DNA or RNA. on the basis of strand, whether it is single stranded or double stranded, on the basis of sense, whether it is positive sense or negative sense. Positive sense means, suppose positive sense RNA, at this case such RNA is no need to transcribe into a mRNA, it get directly converted into a chain of amino acids at time of a translation. So, it is called as a positive sense. Whereas the negative sense RNA, for this RNA there is a need to transcribe into a mRNA and then it is ready for a translate. Method of replication, whether it replicate into a nucleus or a cytoplasm. These were the criteria used by David Baltimore for his classification, and Baltimore divided the viruses into 7 groups. The first group shows a double stranded DNA viruses, second single stranded positive sense DNA, third group double stranded RNA viruses, fourth group single stranded positive sense RNA. In a fifth group he were put single stranded negative sense RNA containing viruses. Sixth group he put the single stranded positive sense RNA with DNA intermediate, and in a last 7th group double stranded DNA with a RNA intermediate. These were 7 groups given by Baltimore for a virus classification. Now, in detail, the first group, that was the double stranded DNA containing virus. It includes the viruses like adenoviruses, herpes viruses, poxviruses. Some of them replicate in a nucleus and some of them replicate in a cytoplasm. For example, the adenoviruses, these viruses replicate in a nucleus of a host, whereas the poxviruses replicate in a cytoplasm of a host. Second group, single stranded positive sense DNA. The single stranded positive sense DNA viruses, firstly they forms a negative sense strand and their replication occurs in a nucleus, for example parvoviruses, and this is an exception for rule of a DNA. We know that all the DNA has a double strand, but the genetic material of some viruses having a single stranded DNA, example parvoviruses. Then the third group, double stranded RNA containing viruses. The double stranded RNA shows a segmentation, and each segment or each genome is transcribed separately and which produces a monocistronic mRNA. Example Pirnavirus and revoviruses. Then the fourth group, single stranded positive sense RNA. This group contains a positive sense RNA viruses. It may be a polycistronic mRNA, for example picornavirus and a hepatitis A. Complex transcription, example toga viruses. Then the next group, negative sense single stranded RNA containing viruses. In this group, this RNA may be segmented or non-segmented. Segmented orthomyxoviruses, non-segmented rhabdoviruses, for example rabies virus. The next group, single stranded positive sense RNA with DNA intermediate. Such kind of RNA does not serve as a mRNA, and it makes the template, and this template further get reverse transcribed into a DNA. Example, that is a retroviruses or a HIV. Then the next group, double stranded RNA with a DNA intermediates. Double stranded RNA with a DNA intermediates, hepatinoviruses. In this image, you can observe the 7 groups of a Baltimore on the basis of genetic material, strand, whether segment or non-segmented, or whether it is positive sense or negative sense. So, immediately take a screenshot of it. Then the ICTV classification for viruses. ICTV stands for International Committee on Taxonomy of Viruses. It was set in 1970 and it includes 9 orders, 131 family, 46 subfamily, 803 genera and 4,853 species. The ICTV, this system of a virus taxonomy, uses a taxonomy which is used for a classification of cellular organism like plant, bacteria and animal cell. And we know that a cellular classification, it has a taxon. The taxon suffixes different words where the order always ends with a suffix that is a virals. A name of family always ends with a suffix viridae. Name of subfamily ends with a Phagoviridae, and name of genus, it ends with a name that a virus. The ICTV classification, it has 9 orders: herpes virals, caudovirales, ligament virals, mononigavirales, nudovirales, orterovirales, picornovirales and thymovirales, and 9 one is bunyavirales. The first order, that is herpes virals, and this order contains double-stranded DNA viruses which cause infection to a eukaryotic organism. That means in a herpes viral order, bacteriophages are not included, whereas it contains only a double-stranded plant viruses and animal viruses. Caudovirales, it consists of tailored double-stranded DNA bacteriophages. Third order, ligament virals, it contains a double-stranded linear DNA containing viruses, and particularly these viruses having a host, that is a arching bacteria. Then the fourth, mononigavirales, it consists of a single-stranded RNA plant and animal viruses, and this single-stranded RNA is monosegmented and negative sense RNA. Then the fifth order, nudovirales, it consists of positive sense single-stranded RNA viruses which infect to vertebrates and animals. Sixth order, orterovirales, this order contains single-stranded RNA and their intermediate is a DNA, and after formation of intermediate DNA with the help of reverse transcriptive enzyme, this DNA replicate and causes a multiplication of viruses. Then the seventh order, picornovirales, it consists of small positive sense single-stranded RNA viruses and it causes infection to plant and animals. Then the next order, thymovirales, it consists of monopartite positive sense RNA viruses which cause infection to plants. And ninth order, that is bunyavirales, it consists of tripartite positive sense single-stranded RNA viruses. These were 9 orders of ICTV classification. So, thank you very much. Please like my video, share it to your friends, Subscribe my channel Easy Life Science and press the bell icon.